This paper also showed that glutathione is dramatically increased, nearly fourfold, by treating cells with Protandim. And it's not because Protandim contains the precursors you need to make glutathione. It doesn't. How's it working? It's causing the enzymes that make glutathione to be produced. That's the part that's rate limiting. The mechanisms are very different. This shows how cysteine, one of the key amino acids in glutathione, gets into your cell, and the enzyme shown there in blue, glutathione synthase, is upregulated by Protandim, so you can make GSH, glutathione, an important antioxidant protectant in your cells. But amazingly, what limits the amount of glutathione most cells can make is not the amount of cysteine that's circulating through your body. There's a little protein there called XC-, not a catchy name. And it's a turnstile protein. If your cell is like your local branch bank, it may have a revolving door to get into that bank. And this protein lets cysteine get in one side of that revolving door, but only if another molecule, glutamate, is coming out the other side of that revolving door. So it's like a heavy door that it takes two people to push to get one in and one out. That's one of the most powerfully upregulated genes by Protandim. And that's the part of this whole of this whole glutathione synthetic pathway that's limiting. That's the slow point. You may have cysteine lined up outside the bank waiting to get through. If you don't have a working door or enough doors in that bank, no matter how much cysteine is waiting outside, it's not going to happen. All right? So making glutathione is not getting more cysteine into your body or acetylcysteine or any precursor. It's about upregulating those enzymes that can put it together and make it for you. This paper is one of two, the third paper, that looked at this standard skin cancer model studied by scientists in the laboratory. It's done in mice, and it involves the application of two chemicals to the skin of the mouse. The first causes mutations, damage to the DNA by free radical mechanisms. And the second chemical is a tumor promoter. Cancer, fortunately, is a multi-step process. If cancer resulted from a single mutation, we would probably all be dead long ago from cancer. So cancer takes a series of mutations in a specific set of genes to cause your cells to lose control. They lose the constraints. It's actually easy for a cell to eat food and divide. The hard part is for your body to keep your cells from doing that day in and day out. Many of us in this room don't weigh any more than we weighed a year ago, even though we've eaten a ton of food in the interim. And that's a good thing, because if every cell in your body had unrestricted ability to divide and divide and divide, we would all fill this auditorium individually. So it's much more difficult to keep your cells in control than to let them just proliferate ad nauseum. And so cancer happens when those constraints are broken, when your genes can no longer keep a cell from dividing and dividing and dividing. So once a cell breaks those constraints, it is medically a cancer, and it will grow until it consumes you. This paper concluded overall, the induction of antioxidant enzymes by Protandim may serve as a practical and potent approach for cancer prevention. A follow-up paper, about a year later, looked further into the mechanism and showed two of the specific genes that are regulated by Protandim. One is the suppression. Protandim, some genes are turned down, and p53 is one of them. And manganese SOD, that's a mitochondrial SOD, was induced, is turned up. And it was those modulations of those two genes may play an important role in the tumor suppressive activity of Protandim. What are the data shown in that paper? Those two papers. There were two groups of mice, and the first group got a regular mouse chow diet. It's what Purina makes and what mice eat. And it's balanced, it's a healthy diet. When they were treated with the two chemicals at high doses that cause skin cancer, that group of 15 mice, shown here in the yellow bar, got cancer. 100% of them, all 15 mice, developed cancers. Many of them developed, probably all of them developed more than one. The number of cancers per mouse, 6.3. The total number of cancers in that group of mice, 94. The second group of mice treated identically, except their mouse chow had Protandim added at a dose calculated to be equivalent to what most of you in this room are taking, 1 caplet a day for a human, same adjusted dose for a mouse. And look at the second group, 15 mice. This time, a third of them developed no tumors at all. And if you look at the total number of tumors in this group compared to the other, it's 57% lower, only 40 as opposed to 94. So the cancer incidence was reduced by 33%. The multiplicity, the number of individual cancers, reduced by 57%. This was a remarkable study described in these two papers. This is paper number 6, published last year. Virginia Commonwealth University. A very complicated model, but one that resonates with what you heard earlier this afternoon. This is a conclusion from the abstract. Protandim prevented fibrosis and capillary loss and preserved heart function, in this case, right ventricular function, right side of the heart, in this particular model. How did it do that? It prevented heart fibrosis. I told you Protandim turns down some genes and turns up others. There are genes that signal the fibrotic process, formation of scar tissue. And again, if you have a cut, it's important that you can heal that cut. But if your heart turns to scar tissue, you're in big, big trouble. The graph on the left shows the amount of fibrosis in the hearts of these animals. They were treated with, here, a chemical that caused pulmonary hypertension. That's also a word you heard earlier this afternoon. Pulmonary hypertension puts a strain on the right side of the heart that pumps the blood to the lungs to get reoxygenated back to the heart, and the left side then pumps that reoxygenated blood to every part of your body.